So all set, should we start? Hello?
Hanji.
Should we start?
Yes, please.
Just give me 1 minute. Let me start the YouTube. Yeah, we are live on YouTube, so we are starting. Good evening, everybody. Welcome to this evening episode of Pursue. This is Pursue 14G, which is Hematology General and Fundamental. We are streaming live from GMCS Chandigarh via Kolkata. We have a very important person on a very pertinent topic. The topic of the day is structure of the marrow and the hematopoietic microenvironment, which is the basic fundamental of hematology. And to talk on that, we have Dr. Anita Tahlan. She is an MD, DNB, MAMS, and a DM in Hematopathology from PGI Chandigarh. Presently, she is a professor in the Department of Pathology at the Government Medical College and Hospital, Chandigarh. She was awarded the Prof.R.Nath Gold Medal for Best Published Research in 2000 at PGI Chandigarh. She is a reviewer and referee of various specialty journals with multiple publications in national and international journals and book chapters. Before I ask Dr. Tahlan to start, let me request all of you to please keep your mic muted, your camera off, and please don't share your screen. With this, let me request ma'am, please share your screen and let's start.
Is it visible and am I audible, Dr. Nadi?
Yeah, just press that hide, ma'am.
Yes.
You're perfectly fine. Please start. Thank you.
Thank you so much, Dr. Nadeem, for giving me this opportunity. Today I'm here to talk about the structure of bone marrow. Over the years, various new modalities have developed, and initially bone marrow used to be very painful, but now with recent advances, the procedure has become better and more comfortable for the patient. Therefore, it is absolutely pertinent for us that we should be able to glean as much information as we can from this invasive procedure. I shall be talking about both the bone as well as the bone marrow. Bone marrow is the soft viscous tissue which occupies within the bony cavity, and it is composed of a heterogeneous population of cells which are either directly or indirectly involved in the primary function of hematopoiesis. It is implicated in almost all the diseases, and it is also one of the favored sites for metastasis. And let's see how it influences our aging. So bone marrow improvements have also made us see the bone marrow more carefully. However, it is difficult to analyze the bone marrow because it is enclosed within the bone, and therefore it is not available for imaging studies. And a majority of the studies which are done on mouse marrows. So marrow is one of the largest organs and constitutes around 4 to 5% of our whole body weight. And its main function is that it produces the hematopoietic cells, which are supported by the stromal cells, and then these cells, they mature and they are released into the peripheral blood, where they perform various functions. And the daily workload of bone marrow is around 2.5 billion red cells, 2.5 billion platelets, 1 billion granulocytes per kg body weight, and it has to keep adjusting from the neonate or intrauterine life to a 70 or 80 years old male. So all this time, various adjustments need to be made. Hematopoiesis occurs in the cavities of certain bones that contain hematopoietically active red marrow, and whole of our skeleton does not have this active red marrow. And initially, the bone formation is predominantly endochondral ossification, which occurs in the long bones. So the cortex of the bone is lined by the periosteum, which are lined by the periosteal cells. So this cortical bone is very thick and encompasses the bone marrow. Then this cortical bone then gives a honeycombing of these bony trabeculae, which are lined by the endoosteal cells, and they run parallel, and in between they have the cement lines where they get mineralized and ossified. So you have the periosteum, which gives the strength and the shape, and then you have this predominantly parallel trabeculae, thin trabeculae. So an anastomotic network of thin trabeculae is there, and in between this anastomotic network, there lies the semi-solid viscous bone marrow. Now, the bone marrow is not uniformly present in the medullary space. There are these clusters of stem cells and their progenies, which lie as irregular cords. Although they may be appearing irregular cords to us at present around the sinusoids, however, they are absolutely spatially arranged, and they have a specific purpose why they are present in that location specifically and not some other location. So the spatial arrangement of the bone there is a constant modeling of the bones, and there is significant structural and functional heterogeneity in our skeleton. Therefore, the bone needs to keep interacting. What are the cells which are necessary for doing this? We have the osteoclasts, which are responsible for the reabsorption of the bone. We have the stem cell and the osteoblast, which form the bone matrix. Osteocytes are present where they maintain the bony tissue. So starting with the osteoblasts, their main function is formation of bone, regulation of bone rhythm, CSF, IL-1, IL-6, and they also produce various cytokines like TGF, osteopontin, and CXL12. I shall be talking more about this and their role a little later. The other cells we have is the osteoclastic cells. Osteoclastic cells are the multinucleated giant cells, the stem cells from the marrow. They communicate with the osteoblast via a rank system, which we shall talk later. So this RANKL and OPG system of signaling is the one which helps the osteoblast and osteoclast to interact. So therefore, the bone remodeling is continuous, well regulated, and there is significant structural and functional heterogeneity in the sense that in adult marrow, only the flat bones will have the red marrow, the rest of it will be not there, whereas in a neonate, the active marrow is present at almost all places. Now, this bone remodeling is basically done at basic multicellular unit. Remove the mineralized bone, which is done by the osteoclast. Bone formation is done by the osteoblast, which is followed by the mineralization. So both bone and the bone marrow, they function together in this regulation. Now, where do these BMUs reside? So these BMUs, they are done, this work of BMU is done in what is called a bone remodeling compartment. So what happens is that there is a canopy which is formed, such as here. So in this canopy, the lining cells and osteoblasts will come and form the new bone, and then it will get mineralized. So we have the bone remodeling compartment where this bone basic multicellular units, they do their function in the bone remodeling compartment. So at a given time, all these processes go on, maybe at different sites in the bone marrow. Usually the initial event is the reabsorption. However, formation and reabsorption, the cycles keep going on. There are how the bone is influenced by various pathological states is well evident by the bony changes seen in severe anemias like beta thalassemia major or sickle cell anemia, where the bone has got modified, and it shows here on end appearance there is widening of the facial bones, and in sickle cell H-shaped vertebral bodies are seen. So these prove that whenever something is happening in the marrow, it also influences the bone itself. Other examples are the systemic mastocytosis and Gaucher's disease. The main supply of the bone marrow is by the nutrient artery, which is the main supplier. A less important supplier is the periosteal artery. This nutrient artery gets divided into radial branches. These radial branches go and pierce the cortex, and then the blood from this and as well as the periosteal arteries mixes, and small capillaries are formed here, and these capillaries join to form sinuses, which then convert to venous sinuses, and then these venous sinuses combine to form central sinuses, and the blood goes back through the emissionary vein. So these sinusoids, this heavy sinusoidal shows a little less concentration of oxygen and maybe hypoxic also. What about innervations? So piercing the bone and then going into the bone marrow to innervate is done by both myelinated and non-myelinated fibers, but it is not done very frequently. So these small twigs, both myelinated and unmyelinated, non-myelinated, they elaborate various hormones, neurohumors, which will affect the hematopoiesis. Made of nerve terminals, which are adjacent to the periarteriolar adventitial cells. Now, periarterial adventitial cells themselves, they make a syncytium. So once they make a syncytium, even if there are small nerve fibers, it gets amplified and the job is done. So this was in brief about the bone is vascular and its neural components. Now the other two main components in the bone marrow are the hematopoietic system and the subacute tissue. When we talk morphologically about bone marrow, we talk about these couple of terminologies. I'm sorry, which are the bony trabeculae, and the area around the bony trabeculae is called a paratrabecular region, and in between we have the interstitial area. So when we are describing morphologically, we talk in terms of paratrabecular, sinusoidal or interstitial localization of the cells, the stroma or any other tissue that we are talking of. So hematopoiesis occurs in the extravascular space between marrow sinuses, And the cellularity is obviously more in a 3-year-old child, active marrow, rest would be fat. Now these hematopoietic cells are not arranged in random, but as I said earlier, they have consistent spatial relationships, and the main hematopoiesis occurs when this is the, let's suppose, an endothelial cell. This is the luminal side and this is the abluminal side. The luminal side, the abluminal side has a coat of adventitial reticular cells. The syncytial cells of neuroreticular complex that I talked about is also here, which is formed. So these hematopoietic cells, they migrate into the extravascular space on the abluminal side, and they proliferate and differentiate under the influence of the non-hematopoietic cells, which are the stromal cells, and the extracellular membrane. They help and direct its proliferation and differentiation. So what is the spatial organization that I have been talking about? So erythroid series are located in the center intratrabecular region, as well as the megakaryocytic series, whereas the myeloid series are located in the endosteal region. So what do we want from our erythroid series? That they should produce almost million cells so that they meet our requirements, and this makes the erythropoiesis a very complex and highly regulated process. And these precursor cells, they undergo various series of structural and biochemical changes under the effect of cytokines and transcription factors like erythropoietin, GATA, EKLF, and SL. And we also want that whenever there is a need, this biological process should get accelerated, and it does get accelerated, and our levels are maintained in a normal physiological range. So morphologically, as I said, that they are present in the intratrabecular region or the interstitial region, and they can be identified on a H&E section by these darkly staining, which are called the ink dot nuclei kind of cells, and which is a large cell. Then we have intermediate and late erythroblast, which later gives rise to a anucleated RBC. Now this central macrophage Pearl stain will demonstrate that it also has iron, and these are the various stages of maturation. These cells, they look dark blue. You can identify the macrophage, some debris is there in the macrophage, and the various erythroid cells are surrounding the macrophage. So this is called an erythroblastic island, which is the basic characteristic unit for a hematopoiesis. Now, what does this macrophage do? It can be similar to old lady sitting in between and feeding the kids around. And when she is feeding them, she is also trying to give them stories, or the kids are enjoying themselves around a central table. So if we extend the same thing, then around a central macrophage, these erythroid cells are sitting. So let's see what are these erythroid cells. So around the macrophage, the early erythroblast is sitting on the macrophage. As it matures, it needs to extrude its nucleus. So the macrophage eats its nucleus, and then the reticulocyte gets detached from the macrophage. So that means the erythroblast is giving anchor and forming an island for these cells. It is also providing various proliferation and differentiation interactions. So what are these interactions? We know that we have various extruded nuclei, and obviously we know about the transfer of iron when it is attached to the macrophage. So in a healthy state, the erythroblastic island will provide iron that will also promote erythropoiesis and enucleation by these membrane interactions and will eat up the phagocytosed nuclei. What happens in stress and disease? In stress and disease, for example, whenever there is stress, the splenic macrophages will engulf these RBCs and produce anemia. And depending on the disease, the macrophage may be depleted or may be increased. So just to say that this erythroblastic island will respond to the disease process. So what are we supposed to look for in the erythroid series? We have to look at the proportion of erythroid cells. We have to look at the various relative proportion of these, especially the morphology of erythroblast and its erythropoiesis if it is there. Coming to the next series is the megakaryocytic series. Megakaryocytes are fairly rare. I would say rare because they are almost 1 in 10,000 cells, which is a megakaryocyte. It is a large cell and produces platelets by fragmentation, and the production may increase even 10 to 20 times, and we use various thrombomimetic drugs which increase its production. So the megakaryoblast is a small cell, but the megakaryocyte that we are talking about is the largest cell in marrow, which is around 50 to 100 micrometer, and it has got these long pseudopods which ultimately give proplatelets or platelet formation is there. This occurs because of endo-reduplication which occurs in these. So where do these megakaryocytes sit? So they sit in the intratrabecular region, but where in the intratrabecular region? They sit in the abluminal side of the sinusoids. On the abluminal side of the sinusoids, say the proliferate become larger, and they sit on these adjacent to the sinusoid. Adjacent to the sinusoid. When they are sitting here, they give pseudopods. They give these platelet clumps, platelets which are discharged into the sinusoid. It's just like a long extending your hand and giving out something from your fingertips, where these platelets are discharged in the sinusoids. Another purpose these megakaryocytes do is that some cells which are traveling, for example, a neutrophil wants to get released into the system, so it can go through the megakaryocyte and get released into the circulatory system directly. This we have been calling as emperilopoiesis, where these big megakaryocytes are abutting the sinusoids, either in the perisinusoidal or in the intrasinusoidal places. PA stain can be used to highlight this. So what are the localization and their clustering, if any? What about myeloid series? The myeloid series is located at the paratrabecular region or the endosteal region. So this is the paratrabecular region where the myeloid precursors are located. They go towards the intratrabecular region, where in the interstitium you have the maturing myeloid series. So immature myeloid series would be in the paratrabecular region, and mature myeloid series would be in the interstitial region. If the immature myeloid series comes here and can be seen in MDS. So for myeloid series, we are supposed to look at the proportion of granulocytic series, their relative proportion, morphology, and ALIP if it is there, or any other signs of dysbiosis. Lymphoid cells are not so many in the marrow, and they are usually scant. However, sometimes they can form aggregates, and reactive lymphoid aggregates almost never have a paratrabecular location. So if you find a reactive lymphoid aggregate in the paratrabecular location, they can be held in the perivascular region. So plasma cells are located in the perivascular region. CD138 positivity can highlight these cells. So in addition to the environment includes the vascular sinuses, the extracellular matrix, the stromal elements, and these are the cells which provide a structural scaffolding where the cells reside. So this is one cross-sectional diagram where we have the nutritive vessel, the cortex, the nerves, and the reticular cells, sinusoids, and the parenchyma. So the stroma will consist of the vessels. The red will discuss these in some detail. Majority of them are supposed to arise from this skeletal stem cell, which is located on the bone marrow sinusoids. And there is significant transdermal plasticity in the sense that these cells can be formed whenever they are required. So what are these cells? So the various cells which are included in bone marrow stromal cells are the adipocytes, which are in the central core, and yellow marrow. They indirectly and directly also influence the hematopoiesis. Endothelial cells can be seen in the sinusoidal lining or in the arteriolar lining, and these are the entry to the bone marrow, and therefore they enable the exchange of various molecules between blood and marrow. Fibroblasts, they synthesize the reticular network and the collagen at times. Osteoblasts are usually present on the cortical region, and they synthesize bone tissues and regulate the bone angiogenesis. And osteoclasts and chondrocytes, they. In addition, the stromal cells also nerve growth factors like BCAM, tensin, endoglin, collagens. They express IL-6 receptors, IL-1, insulin-like growth factors, and various chemokines. And they also express TGF-beta c-KIT, which is a negative regulator of hematopoiesis. So stromal cells may promote, as well as the luminal side is lined by the endothelial cells, and then there is a basement lamina, and then these cells and the abluminal side, the adventitial reticular cells combine to form a meshwork. So that is how the sinus is organized, and this is the main barrier, and this is where all the interchange occurs. It is actively endocytic, and it expresses VWF, collagen, laminin, various adhesion molecules. So because it is one of the main barriers, therefore it is capable of regulating the cellular traffic. Adventitial reticular cells, which are on the abluminal side, they synthesize the reticular fibers, their cytoplasmic processes, they form a meshwork, and they constitute the reticulum of the marrow, and they express CD10, 13, and class 1 antigens. In disease states, we have seen, especially in myelofibrosis, the vascular architecture gets disturbed, and you can see dilated sinusoids. Fat is an integral part of the bone marrow microenvironment, and the bone marrow fat results from an accumulation of fat cells within the marrow. And as the age increases, a big lipid vacuole of triglycerides, which is made up of fatty acids, which can be saturated, mono or polyunsaturated. So initially these adipocytes were only thought to be filler cells, which just filled the void, which was left by the hematopoietic cells. But now, as our understanding is increasing, we know that they have definite roles. So they are the major sources of energy, and they are also modulators of the adipocytes. They can provide local energy. They can also influence the osteoclast and influence the osteoblasts. They secrete adipokines and leptins and influence the mesenchymal differentiation cells, and they are also implicated in malignant cells. So this we have seen earlier that whenever we give steroids, there is osteonecrosis, especially of the femoral neck. So when these adipocytes they undergo ischemic, they might lead to osteonecrosis. So is it the same fat that we have at other places like subcutaneous tissue and visceral tissue, or is it something else? Probably it is something else, because this is not localized in lobules, but it is scattered. It is much smaller in size. It develops from the mesenchymal stem cells. In addition to storage cells, it also acts as a secretory cell with leptin and adiponectin. And it doesn't undergo lipolysis. It promotes hepatogenesis and osteogenesis. It expresses rank, which promotes osteoclastic differentiation. And overall, it has an important role in microenvironment which is being studied. What about extracellular matrix? The mesenchymal cells, they form a stroma, and they are actively laying down extracellular matrix, and proteoglycans, fibronectin, genexins. They are all a group of macromolecules which are distributed on the surface of adventitial cells. They help in cell-cell interaction, cytokine presentation, cell differentiation, and various interactions like collagen, laminin, thrombospondin, they all help in interactions. Now coming to the main cell, which is the hematopoietic stem cell. So what do we want from this hematopoietic stem cell? We want that it should produce what we want and it should stay quiet when we don't want it. So in some words, we want it to be multipotent so that it can give rise to multiple cell types. It needs to keep itself for a long time of span, so it should be have long-term renewals. And we have the niche where it should be dependent, and we should be able to maintain the cell in an undifferentiated state in the niche. Once it leaves the niche, then it gets differentiated, but in the niche it remains undifferentiated. And it should also have a property of long-term repopulation. So what we want is that long-term it should be capable of self-renewal. Short-term it can go out, produce various kinds of cells, and then depending on our needs can do that. When we were residents, we were taught this, that the hematopoiesis, there's a hematopoietic cell which gets divided into a common lymphoid and a common myeloid progenitor, and then this myeloid progenitor gives rise to megakaryocytic, erythroid, and granulocytic progenitors. So there has been some advancements in this, and a refined model of hematopoiesis is suggested now, where it is thought that instead of this three-tier mechanism, probably the hematopoietic cells are many and they directly get differentiated into various cells. And megakaryocytes get differentiated much early. So this is called the evolving hematopoiesis concept, which says that there is some revision in the classical hierarchy as several new cells are identified. And instead of the three-tier, there is a continuous differentiation which is occurring, and probably the megakaryocyte lineage segregates early. And this heterogeneity probably reshapes the balanced differentiation tree map. So instead of this discrete differentiation, probably there is a continuous differentiation landscape which is more favored now. Now these hematopoietic cells, they need to be preserved in their niches. The concept of niche was first proposed by Sheffield in 1978, who said that there is a physical existence of niche in the bone marrow, and there are a variety of cells which make that niche and which maintain that niche. As I said earlier, it is difficult to image the bone marrow because it is located in a cortical bone and therefore inaccessible for imaging. But the newer tests have somehow added to our better understanding. So what are our requirements from the niche is that we wanted to tightly regulate the number of stem cells, and the constituents of the niche should be able to stimulate or inhibit the stem cell number and function. And physical interaction which occurs between these different type of cells which ultimately maintains the stem cell in its state. So these are together called SMART properties, which are self-renewal, maturation, apoptosis, resting, and trafficking. So these SMART properties are the ones which are required. So in a nutshell, niche will have a cellular microenvironment and a non-cellular microenvironment. The cellular microenvironment will have hematopoietic cells and the non-hematopoietic cells, and the non-cellular microenvironment will have the extracellular matrix which we have discussed. Non-hematopoietic cells, the predominant cells are the endothelial cells, the fibroblasts, and the cells involved in bone hemostasis like osteoclasts. And in apart from the various series, the various immune cells, the bone-derived circulating endothelial precursors and progenitors are also included here. So where are the niches that we are talking about? As I said earlier, the osteoblasts, they contribute to hematopoietic stem cells, so they can be one candidate. Other can be the endothelial cells because they are the barriers between a developing cell and blood. So therefore, these initial sites of entrance are controlled by endothelial cells, so they are very important. And third is the perivascular side. So osteoblasts, endothelial cells, and perivascular sites are the niches. So these are well recognized sites of stem cells now, which is the sinusoidal megakaryocytic niche and the arterial parasite. But as we study these, so this is the domicile of the hematopoietic stem cell, both in benign as well as malignant conditions. So when we studied their markers, so then it was found out that different niches have different markers. Sinusoidal niches had more CXCL12, whereas arteriolar niches had PEA cells, which I had described in neuroreticular complex, and similarly for these cells. So it just goes on to say that somehow all these niches that we talk about are different from each other a little bit. So the osteoblastic niches, the reticular niches, and the perivascular niches. So all these niches, they have one cell. So which are the cells which constitute? Probably CXCL12 positive cell, because it's seen in osteoblastic, reticular, and the perivascular niche. In addition to these CXCL12 positive cells, we have nestin positive cells, we have Leb R positive cells, and we also have Schwann cells. So out of all these cells, CXCL12 positive reticular cells are the one which are more frequent, and they are the one which are so CXCL12 represents the homeostatic mechanism, a chemokine, and it is produced by various cells in bone marrow, and therefore it is implicated in maintenance of the hematopoietic cells, retention of the hematopoietic cells in its niche, and also maintaining and inhibiting the uh, hematopoietic cell from going into the cycle, so therefore keeping it quiet. There are other niches also which are well studied. So are all these niches same, or is there a hierarchical differentiation, or is there a hierarchical niche which is first, which is second, and are they specific? So this hemopoietic retention in the bone marrow and mobilization to the circulation is regulated by a joint action of chemokine, hormone, neurotransmitter, and osteoblastic interactions. So it's very dynamic, and probably these niches, they have different roles, but it is not very clear at present. What about its neuromodulation? So the immune cells in a steady state, the CXCL12 and CXC4, they anchor and keep the hematopoietic cell in its niche. But whenever there is an external factor, which can be in the form of GCSF, These CXCR4s, they get increased on the hematopoietic cell, the rank gets inhibited, and these cells, they get mobilized and go into the blood vessel. So that means to say that these cells can be influenced by many immune and hematopoietic systems, and this is one GCSF is one molecule which has been commercially and therapeutically utilized for the mobilization of stem cells. Bone marrow microenvironment is affected in both infection, inflammation, benign as well as malignant conditions. And the dysfunction of the interaction between hematopoietic system and the progenitor cells, they lead to different disease states. So what happens in infection is that we see emergency myelopoiesis in the form of increase in left. There is mobilization of mature myeloid cells. These cells go into the circulation, as can be evidenced by the shift to left. And there is hematopoietic injury by the various cytokines which are released in infection. The infection also remodels and damages the microenvironment. So, whenever there is chronic inflammation in the bone marrow microenvironment, it can lead to initiator or even driver of a hematological malignancy. So, all these circulatory cytokines are increased in an inflammatory microenvironment like IL-6, and these in turn affect the megakaryocytes to produce more megakaryocytes. These yellow ones are the inflammatory microenvironments. They affect the erythroid island, which leads to its differentiation defect. In addition, they also affect all these cells which are LEPR positive, nestin positive cells. All these cells get affected, and the hematopoietic support gets decreased, and these lead to myeloid expansion and recruitment of innate immune cells. This also leads to impaired self-renewal and output genotoxic stress, which may give rise to a malignant clone. So there is a possibility of malignant clone whenever we have chronic inflammation. And all this leads to increased reactive oxygen species, which adds further to its insult. So these can lead to malignancies. So in a controlled state, the CXCL12 cell has a hematopoietic cell which is nicely sitting in its niche. But when it progresses to myelodysplasia kind of thing, these cells increase. So do the leukemic cells, they increase. And however, in MDS, they are still controlled, and although they are present at locations where they should not be, but they are still being held to the CXCL12s. Whereas by the time it turns into malignancy, the leukemic blasts have decreased the CXCL12, and they have reprogrammed the cell, and all these cells have increased and are ready to leave the bone marrow. So, in a leukemic bone marrow, although it's highly complex, but various mechanisms are involved. In initiation, various signaling pathways like beta-catenin, JAK1, PTPN11 and ICER1, they are all involved, which leads to proliferation of these cells. When from initiation, when it goes on to expansion and chemoresistance states, it co-opts the mechanisms of the normal hematopoietic cells. So a normal hematopoietic cell utilizes CXCL12 and all. So it utilizes them for its expansion and preservation. After this, it remodels the hematopoietic niche by blocking and causing differential alterations, decreases the healthy hematopoiesis, and is ready to leave the cell. So a leukemic cell initiates, expands, and remodels the whole bone marrow microenvironment system to its favor and later becomes independent of these bone marrow signals. This also leads to the resistance mechanism, which is probably mediated by these cells. In MDS, the increased apoptosis is possibly the most important mechanism, which is dependent on the close cellular contact, whereas in MPN, the cells they become less supportive of normal hematopoiesis because of various inflammatory like cytokines, pre-fibrotic factors are secreted. So the main players here are different, which are the mesenchymal stromal cells, and all this probably leads to more fibrosis and more extracellular matrix which gets deposited in myeloproliferative disease. What about lymphoid disease? So the stromal cells, lymphoma cells also learns to interact with the stromal cells, uh, and both the bone marrow derived mesenchymal stromal cells to its advantage and transforms the bone marrow microenvironment in its favor. What about myeloma? Myeloma is unique in the sense that it is an intramedullary lesion. So this intramedullary cell will affect almost all the cells which are surrounding it, whether it's the mesenchymal stem cells, the endothelial cells, the fat cells, the immune cells, and the osteoclastic cells. One of the main key mechanisms is that the myeloma cells they reabsorb these cells, which is probably by IL-6. Uh, they increase the reabsorption, secretion and reabsorption of these cells. And the characteristically immune suppression and lytic bone lesions are probably caused by the TNF family, which are the rank ligand, and osteoprotein. So this is the rank ligand, osteoprotein signaling that I had initially talked about. Now, this balance between bone formation and osteoclastic bone resorption is disturbed in favor of net bone resorption through increased osteoclastic activity and decreased osteoblastic activity. What about aplastic anemia? The whole marrow microenvironment gets disturbed in aplastic anemia. The defect is in supporting hematopoiesis. There is a problem in formation of these colony-formic units, and there is impairment in physiological maturation and cell surface receptor expression. So on electron microscopy, this is a normal cell where we can see the cells with its stroma. And here there is a scanty degenerative marrow with little intervening stroma, and there are frequent apoptotic holes. So apoptosis is there. Now, a lot of tumors tend to metastasize to bone marrow, and it seems to be the favorite site of many tumors to metastasize. When these tumors metastasize to bone, they upstage their grading. So why is bone marrow the favored site for metastasis? So there is a crosstalk between bone metastatic cancer cells and bone marrow environment. So all these cells which are blood forming and bone forming, they somehow support these cancer cells for metastasis. So the theory that has been suggested is seed and soil theory. So the bone marrow microenvironment gives the soil, and the metastatic cell is the seed. So this seed gets a fertile soil, and therefore it metastasizes. So the various cells like osteoblasts, they help in early dissemination and colonization. They regulate the dormancy. Osteocytes, they promote the osteolytic damage and migratory ability of metastatic cells. Osteoclasts will reabsorb bone and therefore create space for these cancer cells, because bone marrow is a crowded place where we have so many cells. Immune cells will suppress and directly influence the bone remodeling. Adipocytes, they behave like they do in the normal hematopoiesis. Endothelial cells will regulate the dormancy, chemoresistance, and outgrowth of metastatic cells. So all these cells combined, they provide a favorable environment for metastasis to these cells. What about aging? So our hematopoietic cells have divided n number of times and have kept us for a long time. But slowly they tend to lose their ability for self-renewal and regeneration with age. So one of the most important manifestations of aging is the depletion of the stem cell function, which gradually declines inability of the adult tissue specific stem cells to maintain cellular hemostasis of the tissues in which they reside. So when these cells are dividing, they tend to accumulate somatic mutations. And initially they are polyclonal hematopoiesis, which is there in young age, but gradually this polyclonal hematopoiesis give rise to an oligoclonal one, and at some point these oligoclonal hematopoiesis may even become clonal hematopoiesis. This may be one of the reasons why certain malignancies are more in the older age. However, clonal hematopoiesis is not equivalent to an overt pathology. Therefore, just the presence of clonal hematopoiesis should not be considered as an overt pathology. So, clonal hematopoiesis can occur in progressive aging. So, to summarize, bone and bone marrow is a unique system with relevant spatial organizations. There are morphological changes which are identifiable and which are correlated with disease and normal states. Hematopoietic cells are unique in the sense that they respond to all our needs, they keep us going for a long time, and the mesenchymal stromal cells, they keep supporting this microenvironment. The hematopoietic cells, they reside in these special niches, and these niches are composed of a heterogeneous cell population composed of osteoblasts, osteoclasts, adipocytes, fibroblasts, mesenchymal stromal cells, endothelial cells, and all these. And they are well regulated by the effect of growth factors, extracellular matrix molecules. So this crosstalk between the hematopoietic cell and the niche where it is staying is absolutely essential for its survival, self-renewal, migration, quiescence, and differentiation. The newer techniques, whether it's endogenous ablation of a specific cell types or advanced imaging techniques, single cell mass cytometry, they are adding to our knowledge of this bone marrow microenvironment understanding. And various drugs, for example, in multiple myeloma, bortezomib and lenalidomide are used successfully, and they target the bone marrow microenvironment. So bone marrow microenvironment is the topic of current research and targeted therapies. And maybe in near future we shall learn a little more. Thank you.
Thank you so much. Yeah, absolutely right. Bone marrow microenvironment is the area of interest for all the biologists. Thank you so much, Dr. Anita, for a wonderful talk. Very basic but very essential for us to understand the subject. And
Actually, it's a very vast topic.
I would request you.
I thought I should concise it into a way so that we can understand. At least I can understand.
I can also understand. Yeah.
Then it's
hold on.
well done. Sure.
No, it has been well done. Very well, very nicely presented. Excellent. Very nicely compiled. The whole talk was so nice, so relevant, and very, very pertinent. There are no questions on the YouTube. I would request you to share the PDF so that we can upload it on the
I'll
Google
do that.
Meet.
Yes.
Right.
Yes. Thank you.
Thank you
Thank
so
you so
much.
much. Thank you so much, Dr.
Take,
Vidya. Okay, bye
take,
bye. Thank you so
take care.
much.
Bye bye.
Bye.
Good night. Bye. Take care.
Bye. Good night. Bye.